Okay, so let's have a class, and we will move to gram stain today. You already practiced using microscope, and the simple stain, so we'll move to gram stain. So this is lab 3. We'll talk about gram stain. So I'll go real quick about all the principles, since we already mentioned about these in the lab, in the lecture, sorry. So first of all, this is by Dennis Graham in 1800s, which is developed a differentiate staining technology. The differentiated staining technology categorizes bacteria into 2 categories, which is gram-positive, stains blue, or we say stains purple. And we also will have gram-negative bacteria, stains pink, or little, we say red. This is for bacteria. We mentioned in the lecture, for fungi, we all say gram stain blue for fungi. And this is for bacteria. Okay, that's a very simple story behind it. Now, let's move on to today's work. How are we going to do? Lots of the steps are similar. We only have several steps are different. So, procedure. So, first of all, the same thing. You will have a glass slides. But the difference is, instead of using only 1 bacteria, we're going to use 2 bacteria today. So you're going to draw 2 circles, 2 big circles. 1 bacteria you already learned is E. coli. So we will just write EC on the side. That stands for E. coli. Another bacteria is a little bit special. We will learn later, but we are going to be seeing today. It is called E. facile, EF. That is Enterococcus facile. This bacteria belonging to Streptococcus group. So ideally, you will see 2 different bacteria. One is a large, simple large. Another one is a chain-shaped Streptococcus. And this is a surrogate, which is a pathogen surrogate. We did a lot of in our lab. And the detail we'll mention again at exam 2, maybe lecture 3. So that's why I want to introduce you here. So first of all, label. Then second step. Same thing. You flip it over. Okay, so you flip them over. Flip them over, then what's going to happen? When you flip it over, it maybe become EC, EF. So we say flip over. Because we mentioned the Sharpie marker, the dye, could damage the objective lens. So we flip it over. Then we'll move on to the step we call smear preparation. The smear preparation, including you're going to add a big drop of water, then sterilized using the loop to touch the bacteria from the agar plates and then make a smear. Okay, make a smear like that. Same thing like we did for the simple stain. We need to do air dry for 3 to 5 minutes, followed by heat fixing. This is on Bunsen burner back and forth. Okay. Once this is finished, we're going to move on to our gram stain major steps. So what are the gram stain major steps? Number one, we're using crystal violet. We're staining 60 seconds. So crystal violet. Number two, we are going to add in gram iodine. So we mentioned crystal violet is a basic dye we put on the bacterial cell, and the gram iodine is a mordant to make the staining strong.
Sure.
Yes?
That blue color is very hard to see from the dye.
So I'll say crystal violet. Okay. So gram iodine. This is then also 60 seconds. Then the next step is very important. We're using 70% alcohol for only 10 seconds. This is we call decoloration. And I said the 10 seconds is very important. If you overdo, everything could be washed off. Then the last step, we're going to using saffroning. And this is we call also 60 seconds with a counter step. Okay. Following these steps, we need to do a little bit dry, the same thing, and then we're going to do the observation. The observation is always 10x to find specimen, and then move on to 100x with oil immersion. This is we will find 3 important information for the gram stain recording. Number 1, gram reaction. This is simple, positive or negative. Number 2 is cell morphology, which means the shape of the cell. It could be a quark cell, it could be a large, it could be a vibrio. The last one is cell arrangement. It could be tetrade, it could be chain, maybe single arrangement, or maybe cluster like a grape shape. These are the important information we have to record it. Okay. Back to here. Ideally, the results, gram positive, blue, purple, gram negative, pink or red. And we mentioned about 2 theory. What are the 2 theory? If you still remember, number 1 we call cell wall theory. That is because gram positive bacteria has heavy peptidoglycan. So they can hold on the crystal violet color during the decoloration process. That's why the staining stays there as purple crystal violet. The second theory we call it the lipid theory. So what this story talk about is gram-positive bacteria, only 1 to 5% of lipids. In contrast to gram-negative bacteria has 10 to 15% of the lipids. So during the decoloration process, the lipids is dissolved well in the 75% alcohol, it's all washed off. So at that moment, ideally the slide should look like a blank. And then you counterstain with saffroning, it showed you pink color. Because of the cell wall theory, we mentioned about the gram stain, you have to use very young culture. The young culture is usually less than 24 hours, somehow 18 to 24 hours, a fresh one. The reason is we want to make sure the cell wall structure is complete, is intact. If the culture is older, let's say more than 72 hours, then peptidoglycan is start to cracking. Then you cannot hold on crystal violet. At that moment, everything stains like gram-negative. So we say gram-variable stain. So I just say otherwise, if more than 72 hours, we cause gram-variable stain. Another reason possibly cause gram-variable stain, which means you using the decoloration time is too long. So this is the major steps. And I said in the lecture already, the time is not important. 60 seconds, you can do 45 seconds, you can do 1 minute, 30 seconds, that's fine. But the procedure is important, you can't mess it up. Also, the decoloration 10 seconds is important. Okay? So that's pretty much is a procedure. Today, everybody will do individually, have 1 glass slides, 2 bacteria. Again, same as we did before, I'm going to show you how to do a gram stain. Because this is the first time we do gram stain, and this is the first time we're going to use 2 different bacteria. So that's why I want to do an example for you guys. Okay, same thing, we're going to use this bench as an example. And everybody just coming. So we'll see whether we're going to get successful today. Sometimes the staining needs a little bit of luck. And the microscope needs to be working. That's very important. Okay, otherwise, it's all going to be failed at the very beginning. So we'll see how we're going to do it. So the first thing first, I'll borrow 1 rack. We're going to be using new glass slides. You can use the old ones, that's fine. We're going to draw 2 big circles. One is labeled EC, another one is labeled EF. E. coli and E. facile. It looks like this. Okay? Then we flip it over. Now we're going to add the water there. So the same thing like we did before. We want to make sure this is little turn it on. And we also want to make sure the Bunsen burner is working. That works today, very good. So we have a loop, and make sure the loop is straight. Okay, so first thing first, flame the loop, and I said 45-degree angle, sterilize everything. Okay, then what we do, add a little bit of water. So 1 drop, another drop, that's enough. Then we're going to sterilize it again, because we're going to touch bacteria. Okay, so we have 2 slides, 2 plates. First of all is E. coli. So cool down the side a little bit, pick some smear. Okay, let's make E. coli. E. coli is right here. So make a big smear of E. coli. Nice and gentle, and cover the circle. but also make sure it's seen. Now you have to flame it, because we have to kill E. coli, because we are adding E phase sealing. Okay. Cool down a little bit. Want to get E phase sealing. So here, we're going to make a big circle. Okay? So then, flame the loop again. Now we put it on the side. Now let's do an air dry first. The air dry is 3 to 5 minutes. Let's say this air dry is ready. We're going to do a heat fixing, so back and forth a couple of times. Like I said, this is not cooking. So back and forth a couple of times. Okay, you see it's almost dry, and that's ready. Okay. Then we don't need that, so we're going to be turned it off. Then we came here. Don't touch it right now, it's very hot. So we add on the side. Okay, now we're going to move on to the major step of the gram stamp. So we're going to wear goggle. Make sure the goggle is completely cover your eye. And then we're going to have to make sure our gram stamp reagent is ready. The first time you do, you may not be very familiar. So put them in order, crystal violet, followed by gram iodine. Then we will do 70% alcohol, 95% alcohol, then counterstain with saffron. So everything here is ready like this. And also prepare some of the paper towel for the last step when we do the dry procedure. Okay, so let's get these things ready. First thing, crystal violet. So we're putting them in the middle, okay? Staining 60 seconds. You can count 60 Mississippi or do a timing. Let's say 60 seconds is ready. So we do a very gentle rinse. Very gentle rinse. Then second one, what we do is we're going to add a gram iodine. So gram iodine, we want to make sure it stays strong. Let's crystal violet staining stronger. That's why we call it a mordant. 60 seconds again. Let's say the time is ready. So rinse. Okay, the next step is important. Is for 70% alcohol for staining a decoloration for 10 seconds. So we just put there. We're going to count 10 Mississippi, and let's say it's ready. So you're going to rinse it. Okay, then the final one is a counterstain. Counterstain use saffron. You can add it there. Counterstain with saffron. And then how we stain? We're staining another 60 seconds. Let's say the time is ready. So we're going to add this. Okay. So that is ready. And you can dry a little bit more because sometimes it's very wet. That's ready. Okay, now let's see what it looks like. I followed the procedure, I didn't follow the time. I did it real quick. So I maybe did like 20 seconds, 15 seconds-ish. Let's see what it looks like. Sometimes it works, sometimes it doesn't work. So we'll see. Okay, again, we're gonna practice using microscope. First thing first, plug in. Turn it on, and this is wrong. You never use 4x. That's some people use it. That's an obvious mistake. So we're going to pull down and put the microscope slides in. And then we add an iris diagram at 10x. Make sure it is working there very well. Okay, so we go stage up, stage up, stage up to the place you can move it. So we go stage down, stage down, stage down a little bit. Okay, we should see some very beautiful picture here, right here. Can you please take a look? That's 10x E phase sealing. Take a look. Can you please take a look? That's efficient 10x. Very good. Go ahead to take a look. I'll have 4 people. Okay. That looks fine. Okay, now same thing, we add oil. So one twisted, do not use 40x, another twisted. Then we're gonna add a big drop of the oil. That has to be big. So we add one big drop right here. No oil in this one. So the bottle obviously is all on here. Some of them doesn't have it at all. Okay, maybe that one is good. This one is always good. So we're gonna add one drop. We also could add one drop here. Okay, I'll just take advantage to see what it looks like. Sometimes it works very well, sometimes doesn't work. So we go here, then we say it is called, oh, it looks fine. Make sure iris diagram at 100. Go ahead to see it. That's on the 100x looks gram positive, and the chain shape, and go ahead and look at that. That's 100x is gram positive, purple color. Go ahead to see it. Yeah, go ahead and see it. What it looks like. Gram positive, purple, and it's a chain, coxel, it's a chain. Come here to see it again. I'll have another one. Hopefully it works. Okay. So you can take a picture, which is a very good gram positive cell. Now, we'll just take advantage. Let's just move in to see what it looks like. Okay, I'll move. Where it works, sometimes doesn't work. Oh, we get lucky, so it works. It works, and I'm gonna find a good area, which is you can see some of the pink color there, yeah. But obviously it looks like it's a mixed culture. I think that this salmon, this E. coli may be mixed with some. And so we'll see what it looks like. I don't know. So go ahead to see it. It's a E. coli, what it looks like. It's a large, absolutely different from the E-facing. It's a large and a pink color. But there's a mixed culture there. So go ahead to see it. That's why I say the one is not as good as our lab. It's maybe some different bacteria there. Looks like the baby lactobacillus there, looks like. But it's gram negative. Go ahead to see it again. So you just move around if you focus. Okay, go ahead, come here to see. I have 2 more people to take a look at the E. coli, what it looks like, the gram stain. Certainly not very good. Okay, come here to see it. So you're going to notice it. Some of the bacterial culture in the plates, we didn't do a strict plating, so it's not a single colony. If you pick a cluster, sometimes you get a mixed culture. It's an example of cross-contamination. So you see it's pink, it's gram negative, but you also see something of gram positive, which is mixed. And some are very short long, some are huge, and you will notice that. Okay, so it's very interesting. Then you can take a picture again. You can write in your notebook what the results going to be looks like. If you see a very pure gram positive culture, then you can say, well, that's really good. But if you see a mixed culture, you can discuss the reason may be a cross-contamination. That we'll talk about later on. So what kind of thing talk about called cross-contamination. Okay, very good. So we are finished here. Same thing. We have another thing going to do is endospore stain, where you're going to use bacillus on Thursday. So I'll keep this one here. Everybody do individually. You can pick a new one, but I encourage you to use the older one in your box and use the same procedure to do the gram stain.